we're gonna dig into vision. All right, not just a synthetic Marvel superhero. Instead, it is one of our special senses. What is the stimulus for vision?
Photoreception? Or
So
sorry, light.
light. And what is light?
Photon?
Yeah, photons. I mean, it's kind of a trick question. It's like light is weird. Electromagnetic radiation, like alternating electric and magnetic fields that are pulsing, or you could think of it as particles of energy. So, in general, there's, I think the easiest way to think of it is as electromagnetic radiation. There's the whole spectrum, electromagnetic spectrum. In fact, it's not a bad idea just to remind people what that means. Share. The electromagnetic spectrum, it's basically, again, kind of perpendicular electric and magnetic fields that are vibrating, and depending on how fast they're vibrating, kind of like with sound, we have different pitches, like low vibrations, medium vibrations, really high vibrations. If it's electromagnetic radiation that's vibrating, if it's really low, there are radio waves or microwaves to cook your food, or infrared is little, you can feel as senses heat. There's this range that our eyes detect, and those are the ones that we perceive as visible light. That's kind of the lion's share of the energy coming down from the sun. It's probably why we've evolved to be so sensitive to like distinguishing it well. If it's vibrating even faster, you get into ultraviolet and then up into X-rays and gamma rays, but they're all radio waves, microwaves, visible light, X-rays are all the same beast. They're as related to each other as like low is related to high pitches. They're just the same stuff, but vibrating at different speeds. So in our eyes, we can detect this kind of range. The lower vibrations, the lower frequency we have is red, higher ones is purple. And that's going to be the jobs of our eyes. The rods and cones, specifically the cells, are going to be able to absorb this energy. And it's ultimately gonna open and close channels, which are going to create electrical signals, right? So it's going to be the same kind of thing, but the receptors are going to have to be weirder. The receptors are actually more like antennas that absorb the energy and then create conformational shape changes in receptor. The receptor, instead of binding a ligand, is more like an antenna that absorbs energy. And by absorbing that energy, it's going to activate a G protein and start a second messenger pathway to open up channels and change voltages. So the transduction is going to be a little weirder, but it's still going to be kind of familiar. It's going to be a second messenger pathway mediated by a receptor to change membrane permeability to ions. But instead of binding a ligand, it's going to absorb a packet of energy. And that is going to turn on the G protein. So it's kind of putting that out there. other thing about eyes that is more complicated than the other senses that we've seen up to this point is we're doing more than just phototransduction, right? On the back of the eye and the retina is where you actually have the photoreceptors, the cells that convert light into electrical signals. But we also want something more complicated. We want an image, in which case you actually need to focus things. So we're going to have this whole system with the lens to actually focus the image on the back of the eye. So we don't just see splotches of light and dark, but we see a focused image. We also are going to have the iris that adjusts the intensity of the light. If it's brighter, we can shut down the aperture. If it's dimmer, we can open up the aperture and let more light in. So the eye is going to be both phototransduction in the retina where the rods and cones are, but it's also going to be kind of like a camera where you can adjust the light level and adjust the focus so you get a good image. And we're going to look at both of those aspects of the eye. We're going to look at the phototransduction in the retina, as well as its ability to form a nice focused image using the lens and the iris and stuff like that. So kind of putting that out there. Those are the kind of the two things we're going to be looking at. And actually, a few other things. The eye also is got some interesting physiological challenges because, you know, the lens and the cornea, they're cells, they're living cells. They need nutrients and oxygen, but they also are like have to be clear. So if you don't want to have blood vessels running through them, because then you're just going to see blood instead of like a clear lens. So we have a system of this aqueous humor that is going to be produced and kind of flood around and provide nutrients to the cells and then get drained. So kind of like we saw in the brain, where there's this constant production of CSF and drainage of CSF and problems if something goes wrong. We're going to see that in the eyeball as well. We're going to see this production of aqueous humor and drainage of aqueous humor that's used to basically provide nutrients to these cells that we don't want to have blood vessels to. But if something goes wrong with that drainage, you're going to get a buildup of pressure in your eyeball. What do we call if there's excess pressure building up in your eyeball because that drainage isn't working right? Anybody?
Is it glaucoma?
Yeah, that's glaucoma. So glaucoma is when this system of producing and draining this aqueous humor, this fluid in the eyeball isn't working, and then you build up pressure, and that can damage the retina. It can cause all sorts of problems. You know, when you go in for an eye checkup, you know, obviously they check your focus and things like that, but they also check for your intraocular pressure as well to make sure that that's looking good. They don't just check the optics. They also check, you know, the health of the, you know, the fluid drainage and all that. All right. All right, let's stop sharing that. So that's kind of like the, when there's like a, I hear somebody, I don't know, who is it? All right. So the eyeball, maybe we'll start with that fluid just because we can do it. So here's like an eyeball from the front, in which case you would see, let's say it's, here's the iris, and pupil. So this is supposed to be an eyeball plucked out of somebody's head, just looking at it straight on. The green is the iris. The word iris even just means rainbow. They come in different colors. You know, what is the iris mainly made out of?
Muscle.
Yeah, it's smooth muscle that's going to be able to, in fact, the iris is going to have smooth muscle that has circular fibers. If the circular fibers contract, that's going to constrict the pupil, make the pupil smaller. Which division of the nervous system is going to operate the circular muscle, constrict the pupil?
Parasympathetic.
Exactly, parasympathetic. You know, there are also going to be longitudinal fibers. So longitudinal muscle fibers, and if those constrict, they're going to dilate the pupil. They're going to make it wider. You know, which division is going to activate those longitudinal fibers?
Sympathetic.
Yeah, exactly, sympathetic. So we're going to come back to this when next class, when we talk more about sympathetic and parasympathetic and what kind of neurotransmitters are used in them. We'll talk about what kind of drugs are you going to put in somebody's eye during an eye exam to keep the pupil from constricting when the ophthalmologist shines a light in your eye and stuff like that. So we'll come back to this. What about the pupil? What is the pupil?
It's space, right?
It's
like
just space. It's a hole. It looks black because there's no light on inside the eye, but it's basically just a hole into this hollow ball that is your eyeball. So let us cut the eye in half. We're going to cut the eye open now. And look at it kind of from the side. So this window kind of coming into it is going to be the cornea. Here you can't really see the cornea because it's just the clear window going into the eye. There's going to be the iris. But here now, instead of seeing it as a frisbee with a hole punched out of the middle, it's going to look like a thing from the bottom and a thing from the top, right? And here now you can see the pupil is actually just this space in between that is letting light go into the inside of the eye. Right? If you take a flash picture of somebody, then you get red eye, because then you're actually shining a light into the pupil and illuminating all the blood vessels and stuff inside the eyeball. So the pupil doesn't look black anymore. It looks red because now you are illuminating the inside of the cave. So the only reason it looks black is just because it's dark in there. So the iris operated by little muscles, smooth muscles. There's another muscle, smooth muscle in the eye that we are going to be paying attention to that adjusts the curvature of the lens for helping to focus the image. That's going to be what we call the ciliary bodies or ciliary muscles. Let me just put them in here. It's going to be the lens here. So the ciliary muscles basically can contract or relax and change the curvature of the lens. We'll talk more about that in a little bit, exactly how that affects the bending of the light and the focusing of the image. But while we're here, this is going to be our ability to talk about this fluid that I'm talking about. So the eyeball is very tough. It's got the outside is what we call the fibrous tunic. The sclera is the white, the cornea. You know, it's pretty hard to pop an eyeball, but then we also have stuff inside to help it hold its form. There's the vitreous humor, which is more of a gooey stuff that's kind of filling most of the back. So this stuff I've just drawn here is more of a gelatinous vitreous humor. Vitreous means glass-like, so it's kind of more solid. But the front of the eyeball is just filled with more of a watery secretion called aqueous humor, and it's made by these ciliary bodies. So I should say ciliary body makes aqueous humor. But a humor is like a fluid of the body. It's this in this context, it's an old school definition of humor. Doesn't mean like funny. So these ciliary bodies are making this fluid. This fluid is, it does percolate through the vitreous humor, but more importantly, it's also just flowing around the front of the eyeball. You know, it can nourish the cells in the lens and in the cornea. But obviously, if the ciliary body is making this aqueous humor, we have to be draining it, or else we're going to have a buildup of pressure in the eyeball. So the drainage is through this thing called the canal of schlem, which is located kind of right on this boundary of the iris and the cornea. It's like in this picture, it would be kind of going along kind of more in a donut, right? Because right,
I'm sorry, what's that called again?
it is one of my more favorite names in canal of schlem. There is, trying to remember what it's hold on one sec. Trying to look for its non-eponym name. No, actually, your book calls it the canal of schlem. I was double checking. I mean, a lot of times the tendency in anatomy is not to use these old names that are named after the people who first described the structures for a couple of reasons. One is, you know, schlem doesn't mean anything other than some dude's name, you know, versus like the corneo-iridio canal or whatever. You know, normally they try to name things in a way that makes more sense. And plus there is also kind of the, you know, the DEI kind of thing of like realizing a lot of things have gotten named after kind of these, you know, old white Western dudes who first got things published versus kind of a more realistic sense of, you know, the whole realm of how everything got kind of figured out and discovered. So for lots of reasons, people have been getting away from naming things or using the old names named after people, but canal of Schlem is still used. So if the canal of Schlem is not functioning, you're going to end up with glaucoma. Because you'll be making this aqueous humor, but instead of having its proper drainage, it's going to start just building up intraocular pressure. That pressure can damage your retina and, you know, the phototransduction in ways that we'll talk about later. On the back of the, there's lots more to the eyeball that we're not going to go into because of time constraints, but I will. The other part of the eyeball that we do need to know about is the retina. The retina is the part of the eyeball that actually does the phototransduction. It has the rods and the cones. So like in our lab today, we'll be thinking about the retina to some degree, and we'll be also thinking about the lens, how we actually focus the image on the retina. You know, there are other layers. There's the choroid layer and things like that. We're not going to get into that for this class. Let me look at the time. All right, so what I'm going to do now, I think, is talk a little bit about the retina, since the retina is where the real phototransduction happens. So developmentally, it comes from the diencephalon, the same part of the brain that became your hypothalamus and thalamus and stuff. So it's got a lot of kind of complicated neural structure because it's developed from the brain and then kind of migrates out into the front of your skull. So it's not just photoreceptors. It's actually going to be a variety of different neurons. I'll mention some of their names and briefly allude to kind of what they do, but I'm not going to get too much into detail for this class. So if this is like the back of my eyeball. Here's where the light is coming in. The retina is kind of set up in kind of a non-intuitive kind of structure. The actual cells that do the phototransduction, that convert light into electricity, are actually located all the way burrowed into the back of the eyeball. So rods and cones, we'll talk a little more, we'll talk quite a bit more about them. Rods and cones, these are the things that actually do the phototransduction, convert light into electricity. And we'll talk more about exactly how that works. So these are the photoreceptors. And again, they actually are connected, burrowed to the back of the eye. And then there are a whole bunch of other populations of neurons that make up the retina. I'll name them, but it's not so important that you understand the details of it all. You are going to have to understand rods and cones in more detail. But you know, these then connect to what are called bipolar cells. They get their name because they have two connections coming off of the cell body. There are going to be horizontal cells that connect in. Whoops. The horizontal cells are kind of cool. They do stuff called lateral inhibition. We'll talk, I might talk a little bit about that. The retina is not just about doing phototransduction. The retina is actually going to do some processing to help sharpen up images, enhance contrast, and things like that. So these cells that I'm talking about, you're not going to have to get into a lot of the details, but it's worth realizing that the retina is more than just about turning light into electricity. It does that, but it does more. There's amacrine cells that I'm not even going to draw here, but I'll put their name in there just so you have that word for the future. And then the cells that you do need to know are the ganglion cells. The reason you need to know the ganglion cells is because they have a axon that makes up the optic nerve. And the reason why it's important, why I'm drawing this all out, and like saying that's not so important, all the cells, and yet I'm drawing it all, is because it is important to realize that this backward structure means that where all the axons go, this is going to the brain here. This is creating a spot where you can't have any rods or cones because the axons are taking up all the space. And the rods and cones that are sitting on the back, it's like you can have rods and cones. Red, I want red. You can have them here, you can have them here, but nothing here. So this becomes what we call the blind spot or the optic disc. Gosh darn it. We are going to play with this a little later this morning. But it means that if the light from whatever you're looking at lands on that little part of the retina, that there's no information. We're going to actually see your brain just make stuff up. It's like it doesn't know what is there, so it just like kind of approximates what's likely there and just call it good. And most of the time it's fine, but we can do our little test for the blind spot, which is always kind of freaky. If you've never done it, it's totally cool. So that's the retina. Again, it's sitting here on the back of the eyeball. Like I said, you've got these, ultimately all of these axons coming out the back, which is going to create this blind spot. For the most part, the cells are pretty clear, so it doesn't matter that the rods and cones are on the back, because the light can get through the bipolar cells and the ganglion cells on top of them. The other thing that we need to talk about now is the difference between rods and cones and where you find them. Rods versus cones in this corner, the rods in this corner, the cones. How are rods and cones the same? What do they have in common?
They're both photoreceptors.
There was
Yeah, exactly. They're both photoreceptors. They both convert light into an electrical signal to send to the nervous system. But there's also a lot of differences that you do need to be aware of, that you'll be responsible for. And we'll actually be doing some exercises. I'll be assigning a homework assignment, because normally if we're in the classroom, we can just do it all together and mess around with conditions that will preferentially activate rods or cones. But since we're not all together in a room, we're gonna send you with some instructions for what to do at home so you can play around with them. So, differences between rods and cones. One is rods tend to be active in dim light. There's been experiments showing that you can see a response on a rod with a single photon hitting it. So they can respond to very dim light. Cones, on the other hand, only function in bright light. So if you're walking around on a dark night and just seeing enough that you're not tripping, but not much else, cones are not anywhere in the picture there. Cones only are functioning if there's a lot of light. What are other differences between rods and cones?
cones perceive color more than rods.
Yeah, exactly. So cones can perceive color. Maybe I should just say color vision. Allow color vision. The reason that this works is that there are actually 3 different populations of cones that respond to different frequencies of light. Maybe we should talk about color briefly. Yeah, I think it's gonna, so this doesn't sound confusing. It makes sense to talk about color. So you are thinking about color. Why does my shirt look kind of black?
Isn't it because darker colors absorb light and lighter colors reflect it?
I mean, but no, but my question, well, how come this shirt looks black? So you're on the right track.
Well, isn't it because black can't show a color because it's absorbing all the light, so there's no light to be shown in black?
Yeah, exactly. So it's absorbing the light. It's not reflecting it back. So there's no light landing on your rods and cones. So that's why it looks black, is because there's no stimulus. If now I'm wearing a blue shirt, why does this look blue?
Is it because that's the only color that's being reflected back at you and all the other ones are being absorbed?
So this is absorbing most of the frequencies and then letting the blue reflect back. So the different frequencies that are absorbed versus reflected back are, you need some way to know what frequencies are landing on your retina or not. And the different types of cones let you do that. There are cones that will preferentially respond to blue, but not other colors. So if those blue population of cones are getting excited, then your brain knows, oh, I'm seeing blue. There are going to be red populations, subpopulations of cones. If those are going, then I know it's red. This is always, what about white? What is white?
Reflecting all the colors.
Yeah, so it's a little counterintuitive. Like when you've been in elementary school, you like mix colors, you know, you're mixing pigments. So your intuition of like, oh, yellow and blue make green and red and blue make purple and all the colors together make mud. I'm sure you, I mean, I think most of you must have done that. I remember being so disappointed as a little kid, mixing all the colors together, thinking it was just going to be awesome. And instead it was really not. But with mixing luminance with light, it's very different. If you mix all the colors together, you get pure white light. So it is actually kind of cool. That's why if you take white light and put it through a prism and separate the colors, you have all the colors of the rainbow there in that sunbeam. So let's go back to the rods and cones.
So if somebody was colorblind, does that mean they just don't have that like blue cone to receive?
So
that?
yeah, exactly. So we're going to talk about, that's going to be part of our lab, is actually going to be a test for color blindness. And it's a genetic thing where you don't make one of the subpopulations of cones. So then you don't have the ability to discern that aspect of color. So typically you don't, color blindness is not like complete color blindness. It's like you lose the ability to detect the redness of things. So then things that only vary by amount of redness will look the same. Like brown and green and stuff all look the same to you. So rods, cones. So dim light, bright light. And I should mention, sometimes people get, oh, rods see in the dark. You know, rods don't see in the dark. You can't see in the dark. Dark means there's no light, which means nothing's going to respond. It's just that rods need much less light to respond. You know, so they don't see dark. They see light, but they see very dim light, very low intensity light. So again, the cones have color. If I look at like, you know, frequency of the light and response, I'm going to have for the low, I'm going to have my red cones that respond to these lower frequencies of visible light. I'm going to have my green cones that respond to this more mid-range of frequencies of visible light. And I'll have my blue cones that respond to the highest frequencies. And depending on what mix and match of these cones that you have getting activated, you are going to just notice all the different colors that we can see. So all the colors that we have can be made up with a mixing of these three different luminances. Right, if you've ever looked at a black and white, old black and white show on your TV, but then you look up close, put your nose up to it, you'll realize the pixels on your screen are actually color. They're using red, green, and blue pixels to create the black and white movie. It's just when you step back a little bit, red, green, and blue all together are white. So you can make black and white by just turning on red, green, and blue as white and turning them all off for black. So rods, on the other hand, there's only one population. They're monochromatic. They get activated somewhere around the green, but there's not different versions. So when you're in rod vision, you can tell that there's light or not light, but there's no color. Everything just looks kind of like black and white. Again, you've probably noticed this when you're like at night, you know, the colors are all washed out. You don't see the colors because you're not using your cones, you're using your rods, and rods don't have the ability to discern color. Because the rods are primarily more in the green, people use red as a way to brighten things up without destroying your night vision. Like, you know, a lot of the headlamps that you have now, you can turn on kind of a bright red light to if you need to get a little bit of brightness. But when we're going to talk about rods, rods are so sensitive that when things get really bright, they get what we call bleached. All of the photopigment gets broken down, and it takes a while. It can take minutes, like 5 to 20 minutes for them to really come back online. So like a safe light or these little lights on your headlamps, when they use red to illuminate things, it's going to turn on the red cones, which are going to respond to the brightness, but the rods aren't going to get bleached by red light because they don't even see red light. They're not tuned to absorb that frequency. So that's why the red light on you've probably seen that, the little on the headlamps, you can use a red light to brighten things up without destroying your night adapted vision. All right, other things, difference between rods and cones. Cones are very high acuity, meaning they can detect a lot of detail. That 2-point discrimination is very good. You can have 2 dots really close and still know it's 2 dots. So we say high acuity. Rods are what we call low acuity. It's like, I know there's something on the page, but I can't tell you what it is. Is it a smiley face or a dog or a leaf or a word? I just know there's something there. So low acuity, like I know there's like a hand, but I could not tell you for the life of me how many fingers are on that hand. It's just, I can see there's something there though. So there's something, but you just don't have a detail. Acuity is the ability to discern detail, like 2 different things being 2 things versus one big blurred thing. And then one last big difference is where they're located on your retina. The cones are located in the very center of your retina in this area called the fovea centralis. Just basically, it's like this spot in the middle. Rods are on the periphery of the retina, meaning around the outside. So if I go back, oh, I don't have it. So if here is my eyeball again, I can draw, here's the lem, here's the iris, back. Here was the, did it again. Here's the iris. Here's the ciliary muscles and the lens. So here's my eye. The cones are located kind of right on the very so when you stare right at something, it lands on this fovea centralis on the cones, means it's going to be color and high acuity. So you get to see a lot of detail, you see color. So when you stare right at something, almost all your cones are located right there in this tiny little spot in the center of the retina. The cones are there, the rods are along the edges. Part of the reason why we have the different qualities of rods and cones is just how they're wired up to the other cells of the retina and the ganglion cells. If you look at each cone, pretty much each cone gets its own axon coming off of the retina going down the optic nerve. So that means that you have a lot of detail. If you have cone on, cone off, cone on, I can see that because I have axons for pretty much every single cone. But it also means you need enough light to operate that cone and get it going in order to actually kick it to have a signal going down that axon. The rods, on the other hand, there's a lot of convergence. You have a lot of axons, maybe from 100 different rods all together coming out to one axon, which means that you've got this big collector. You don't need as much light to get a signal. You only, you need much less light because you're collecting over many more cells, but it means you don't know exactly where in space did the signal start from. So we don't have that acuity. We don't have the detail of where exactly did the light land, but we don't need nearly as much intensity of light to actually notice something happened, if that makes sense. So like right now, if you just, the periphery thing, if you put your hands kind of in front of you, back enough that you can kind of notice them, wiggle your fingers around, you'll see there's hands, but you will not see how many fingers are up. As soon as you bring them right in front of your face, it's like, oh my God, I totally see hands with fingers. As soon as they go off to the side, I know there are hands, but I can't tell you how many fingers are on there. I mean, can people see that? Like, put your hand in front and then keep staring straight ahead, but take your hand to the side. You'll see all the detail just disappear. Do you see that? One of the very kind of non-intuitive things about your eyes is they totally suck as a camera. If we were thinking about the, it's like, I have an iPhone 12 Pro with this like zillion megapixel camera. Your eyeball is about a one megapixel camera, which wouldn't even be like a flip phone anymore. It's horrible in terms of what you think of as a digital camera, but it is very good right in this little spot in the center of vision. That's where you have good detail, but everything else, as soon as you get out of that kind of center of vision, is really bad in terms of acuity. I don't think we're going to be able to get into it as much, but there's a lot of kind of amazing research on just how we have to kind of keep reconstructing our visual world, because every time you look away from something, then you look back, you have to kind of like reintegrate it into your view, your kind of model, your brain's eye view of what the actual visual field looks like around you. And you can mess with it really, really dramatically. I mean, if you've seen change blindness, if you've taken a psych class or something, it's kind of amazing how much you can mess with stuff in the world around you. And if it's not right in front of your face, landing on the fovea, you have no idea anything's going on. Yeah, I might show you some of this stuff a little later, because it is just really cool. And I also think a little disconcerting once you realize things you just take for granted about your sensory systems. When you start picking it apart a little more, you realize like, oh, wait a second. I'm like taking a lot of things for granted about my reality that I can't really take for granted. This word fovea is worth knowing also because especially in vision science, it's a verb, to foveate. It just means to stare straight at something and have it land on your fovea, your part of your retina of most acute vision. Again, most acute vision because it's full of cones. If you are stargazing, why, in fact, we'll see, I should check. Does anybody in this Zoom meeting know what you're supposed to do? If you're looking up at the night sky and you're trying to make out a dim star, what's the kind of rule they tell you? We don't think people don't do as much stargazing.
I don't know if this is the rule you're talking about, but I have noticed that if you're not looking at the star, you can actually see it better. Is that what you mean?
That's it. So it might be people don't do it as much. We used to always just go out with our little planispheres and try to recognize everything. And now with like Star Walk and stuff on your iPhone, it's crazy how cool it is to go stargazing.
You just need to find visible stars. It's a little harder to do these days.
Yeah, no, you have to get out of the city is better. But so Catlin was saying, you look off to the side, you don't look straight at the star and you'll actually see it better. Sometimes you look straight at the star and it seems to like disappear, and then you look off to the side and then it's, oh my God, it's there again. Like what's going on with that?
If you're looking straight at it, you're trying to use your cones more, which don't receive light or are not as sensitive to light. So if you're kind of looking away, it would be making use of your rods, which are more receptive to the dim light from a hard to see star.
That's exactly it. So, and it's pretty dramatic. Like next time you're kind of outside at night and remember to, if there's like a very dim star out of the kind of side of your eye and you look straight at it, very likely it will disappear on you. And then when you look away from it, it'll reappear again. Because when it lands on the cones don't respond to it because it's too dim. But when it lands on your rods again, you see it. So that's just something you can do to it's a practical thing if you're stargazing and looking for super dim stars, but it also helps you kind of understand how the rods and the cones are arranged and their different properties as well. Okay. Where are we? 10. Oh, all right. Let's see. Rods, cones. So maybe what I'm going to do, this is something I would like you to do. I'm going to share my screen again. Whoop. Share screen. Okay, we have to get into modules. Firsthand experience of rods versus cones. So this is something that I am going to publish, at the end of the day. It has a little bit of background. There are two types of photoreceptors, rods and cones, and a little thing about visible spectrum, and blah, blah, blah. So I'm kind of talking about a bunch of stuff for you to do. Again, normally we do this in class together. I put down the blackout shades and we all get down into rod vision and back into cone vision. But since we're not doing that together, I think it really is useful to have firsthand experience of what is it really like to have your cones offline and just be using your rods, and then put the rods, bleach out the rods and put the cones back online and see how everything looks different. So this process, you are going to personally experience the difference between cone versus rod mediated vision. Take about 5 to 10 minutes. It's not going to be that long. It will be easier to do in the evening unless you have blackout shades to darken your room, because you have to get things pretty darn dark. Just closing the curtains is probably not going to be good enough unless they're like really blackout shades. You have to pick a room where you can easily create very bright as well as very dim lighting. So you have to have the ability to make it really dim, like so dim, like when you first turn down the lights, you almost don't see anything, because again, your rods are going to be bleached out. It takes a little while for them to come online. But then you also want to be able to flip a switch and then have it all bright again. So the process will be this. First, cone vision. So just turn on the lights, have it bright. Remember that if it's bright light, you're using your cones. And I want you to pay attention to a couple of main things. One is experience of color. How does the color look? Do you see colors? Are they nice? How's your visual acuity? Can you make out detail? Maybe have a book. Can you read the text on a page in front of you? Can you make out how many fingers you're holding up? Like try to look at details. Can you make out details? Second, rod vision. So now turn the lights way down low. So it's like says there has to be a minimal amount of light, but it should be very faint. In this condition, your cones will no longer be responding. And at first your rods will be bleached. So they're not gonna, when you first turn down the lights, you might see nothing because the rods are bleached, and it takes usually about 5 minutes. Waiting 5 minutes is enough time to get most of your rods going online. So what you should do is just set a timer. Do not look at your bright iPhone to time, because that's gonna destroy your rod vision again. So maybe set a timer to just vibrate in your pocket or something. You don't want to actually look at something bright to know what the time is, because that's going to defeat the purpose of dark adapting. So after about 5 minutes, pay attention, look around you again. Just, think about the color. Do you see color? Yes or no? What is it like to experience color? Visual acuity. Can you see fine detail? If you have this book still there, look at it again. Like, you might see there's a book. Is there any way you could make out at all what any of the words say or anything? I want you to really just think about both the color and the visual acuity. And then the third part is back to cone vision. You basically just turn the lights back on and then notice the change. After you've really kind of gotten a feel for the vision in the world of rods, go back to cones and notice how different it is. Again, like, whoa, the colors, whoa, look at the colors are back. Whoa, I can read again. So, and it just says, type a few sentences that describe your visual experiences of color and visual acuity as you went from bright to dim and back to bright. So this should only take you 5 minutes, right? You just have to look around, describe things, turn off the lights, wait 5 minutes, describe things, turn the lights back on, describe things. Done.